and underscores our shared commitment to advancing in our respective fields. I look forward to productive conversations and collective learning as we embark on this educational journey together. SPSR is distinguished for its engaging webinars, further enhancing its status as a leading platform for continuous professional development in the pharmaceutical sector. I am confident that today's session uphold our tradition of excellence, providing valuable insights from a respected speaker. Let us seize this chance to gain new knowledge, participate actively, and progress our field. Welcome, everyone. I look forward to a productive experience for all the participants. Thank you for joining us today. We are gathered today to commemorate the 106th SPSR International Webinar on role of nitazoxanide mediated mitochondrial uncoupling in the clearance of alpha-synuclein by autophagy, implications for Parkinson's disease, a distinguished event hosted by the Society of Pharmaceutical Sciences and Research in association with Malaysian Society for Molecular Biology and Biotechnology. Under the esteemed guidance of Dr. Shashi Alok, the National President of SPSR, the Society continues to play a pivotal role in fostering innovation and advancement in the field of pharmaceutical sciences. The SPSR is devoted to advancing the pharmaceutical field through a variety of strategies. Our mission is to organize conferences that support pharmacy-related initiatives and endorse programs tackling public health issues in India, focusing particularly on disease prevention. We make it a priority to recognize and honor pharmacists whose notable contributions drive progress in our field. At the heart of our mission is a commitment to spreading pharmaceutical knowledge, ensuring a steady and robust exchange of information about pharmaceutical practices and services. By striving towards these objectives, we aim to elevate the quality and impact of pharmacy profession, fostering a proactive and collaborative community dedicated to excellence. It is with great privilege to introduce Dr. Niharika Reddy, Post-Doctoral Scholar, Firestein Lab, Department of Pharmacology and Nutritional Science, University of Kentucky, Lexington, Kentucky, USA, as a distinguished speaker. Dr. Niharika expertise greatly contributes to our event, providing a substantial knowledge and insights. Her participation is certain to enrich our discussions and expands exploration of role of nitazoxanide mediated mitochondrial uncoupling in the clearance of alpha-synuclein by autophagy, implications for Parkinson's disease. We are grateful for her engagement and anticipate an enlightening session. It is our privilege to introduce Mrs. Monika Sabharwal as the convener for today's webinar, who also serves as the founder and National Secretary of SPSR and CEO SPSR Periodicals Private Limited. Her significant involvement and leadership in the pharmaceutical sciences and research communities are indicative of her commitment to the field. We are very grateful to have her guidance in today's session. Now we will start with the short introduction to our guest profile. Dr. Niharika Reddy is a neuroscientist with over 7 years of research experience in the field of neurodegeneration, mitochondrial biology, and autophagy. She completed her PhD at the CSIR Indian Institute of Chemical Technology, Hyderabad, where she developed strong expertise in molecular neuroscience and disease mechanism. Currently, she is serving as the postdoctoral scholar at the University of Kentucky, USA, where her research focuses on the mechanistic role of CYPIN activators in regulating autophagy during neurodegeneration. Her work integrates molecular, cellular, and animal models to investigate the underlying mechanism of neurodegenerative disorders, particularly Parkinson's disease and Alzheimer's disease, with the aim of identifying novel therapeutic strategies. Dr. Amireddy also possesses significant expertise in drug screening and drug repurposing approaches for neurodegenerative diseases. Her research contributions have been recognized through multiple peer-reviewed publications in leading international neuroscience journals. With a strong commitment to advancing neuroscience research, Dr. Niharika continues to contribute to understanding the complex brain disorders and developing innovative therapeutic interventions. It is with great satisfaction and pride that we declare that we have exceeded 1,800 registrations for this webinar. Registrations are received from India, Azerbaijan, Bahrain, Bhutan, Brazil, Cyprus, Ethiopia, Indonesia, Iraq, Kenya, Kuwait, Malaysia, Mexico, Nepal, Nigeria, Oman, Philippines, Romania, Saudi Arabia, Sri Lanka, UAE, Ukraine, Uganda, UAE, and USA. We are deeply moved by the substantial and varied responses we have garnered. Our heartfelt gratitude is extended to each person who has taken a moment to participate in this significant event. Thank you all for joining us today. Let's make this webinar an insightful and enriching experience for everyone involved. We have our esteemed keynote speaker, Dr. Niharika, with us. Welcome, ma'am. Your insights would be highly valuable, and I would appreciate if you could present them in a speech. Over to you, ma'am.
Okay. Hi, Dr. Pradeep. So it's a very great introduction, and I'm very honored to be the speaker in 106th SPSR webinar. So let me share my screen. So can you see my screen?
Right, ma'am. The screen is visible. You can make it full screen.
Yeah, sure. So a very big hello to everyone, and thank you to the SPSR organizers for inviting me to speak in this webinar series. It's a pleasure to be here, and I'm very happy to share my work with such a diverse and engaging audience. As Dr. Shashi Alok told that this webinar has crossed 1,800 registrations. I'm very happy about that, and I really thank Dr. Shashi Alok for this opportunity. And myself, Niharika Reddy, I'm currently a postdoctoral scholar in University of Kentucky. I'm working with Dr. Bonnie L. Firestein. So I did my PhD from India, from CSIR IICT under the guidance of Dr. Shashivardhan Kalivendi. So uh, currently I'm working in the field of neurodegeneration, especially in the Parkinson's disease and Alzheimer's disease. Today I'll be discussing my work on role of nitazoxanide mediated mitochondrial uncoupling in the clearance of alpha-synuclein by autophagy, implications for Parkinson's disease. So it's a key aspect. The key aspect of this study is the concept of drug repurposing, where we explore new therapeutic application for existing clinically approved drugs. And as we all know that there are a lot of drugs repurposed, especially the recent drug that is minoxidil, which were primarily used for controlling the blood pressure, but right now it is used as a topical applicator for hair. So not only that, even metformin, which is a type 2 diabetes drug, now right now it is repurposed to use repurposed to use as an anti-cancer drug. So my work majorly highlights how modulating mitochondrial function can influence the cellular clearance pathway. With that, I'll begin my presentation. So Parkinson's disease. Parkinson's disease is characterized by the loss of dopaminergic neurons in the substantia nigra pars compacta region in the brain. It is the second most common neurodegenerative disorder after Alzheimer's disease and most common movement disorder, with a global burden of approximately 6.1 million patients. So the symptoms include tremor, where shaking of a body part occurs when it is at the rest, postural instability, impaired balance, rigidity, which means stiffness in the muscles, and slowness of the movement. So one of the major prominent pathological hallmark of Parkinson disease is the presence of intracellular protein aggregates, which are referred to as Lewy bodies. The major constituent of this Lewy body is a protein that is alpha-synuclein. So, this alpha-synuclein has been linked to both sporadic and familial forms of the Parkinson disease. Moreover, alpha-synuclein and its aggregation have been widely linked with the neurotoxic pathways, which ultimately lead to the neurodegeneration. Researchers have not been able to pinpoint the specific factor that cause aberrant clumping of this alpha-synuclein protein in diseased condition, which is a fact posing the biggest problem in the Parkinson's disease. So there are various mechanisms involved in the PD, like oxidative stress, environmental toxins, and genetic factors. So increased oxidative stress results in the accumulation of free radicals, leading to the neuronal death. Environmental toxins, including herbicides and pesticides, some of the regularly used herbicides and pesticides include rotenone, paraquat, and the other neurotoxins such as MPTP and 6-OHDA. These uh, toxins inhibit the mitochondrial complex 1 in electron transport chain, causing impaired ATP production, leading to the dopaminergic neuronal cell death. Not only that, the genetic factors, including PINK1, SNCA, which encodes the alpha-synuclein gene, and DJ1, mutations in these genes, particularly in PINK1, causes mitochondrial dysfunction, and mutations in SNCA, widely noted mutations such as A30P and A53T, causes misfolded protein, leading to protein aggregation, and mutations in DJ1 leads to the protein aggregation, ultimately causing Parkinson's disease. So, as what we can see from here is each and every factor is influencing the mitochondria, so that there will be a mitochondrial dysfunction in the nervous system, which is a concern during aging, and it has been associated with major neurodegenerative disorders such as Parkinson disease, Alzheimer's, and Huntington disease. So let us look into the mitochondrial role and how mitochondrial dysfunction leads to neurodegeneration. So mitochondria are essential for neuronal function and survival. As we all know that mitochondria are the cellular energy powerhouses which supply the majority of cells energy in the form of ATP. And ATP supplied by mitochondria powers variety of activities which are essential for the cell survival. So in Parkinson's disease, mitochondrial dysfunction takes place by any of the gene mutations of these genes or environmental factors as we discussed earlier, or mitochondrial mutations. And the major concerning thing is the aging, which causes mitochondrial dysfunction, leading to impaired calcium homeostasis, impaired mitochondrial dynamics, ATP depletion, increased oxidative stress, and neuroinflammation and excitotoxicity, causing alpha-synuclein to aggregate and ultimately leading to the dopaminergic neuronal death. So let us see how these alpha-synuclein aggregates and subsequent death of dopaminergic neurons drives the progression of neurodegeneration. So alpha-synuclein is a single 140 amino acid long polypeptide chain. It is present in multiple brain regions such as hippocampus, cerebellum, thalamus, neocortex, and substantia nigra pars compacta. It is abundantly present in the proximity of presynaptic terminals. But biological functions of alpha-synuclein in Parkinson disease is still a debated topic. However, scientists have identified that alpha-synuclein appears to be involved in a synaptic vesicle regulation, vesicular traffic, and various mitochondrial functions. Sorry. So let us see what are the pathways implicated in alpha-synuclein toxicity. So in the healthy cell, the mitochondrial function and oxidative balance is maintained, and there will be a proper clearance of cargo or debris via autophagy lysosomal system, and endoplasmic reticulum function is preserved, leading to the proper synaptic function and release of dopamine in the synaptic terminals, leading to proper motor functions. But in Parkinson's disease, the mitochondrial dysfunction takes place, leading to the increased oxidative stress, which causes proteins to aggregate, especially alpha-synuclein protein aggregation, where in PD, as autophagic and lysosomal pathway is blocked, causing an accumulation of aggregated protein, which could itself lead to more aggregation. So, causing endoplasmic reticulum dysfunction and synaptic dysfunction in the cells, leading to the less dopamine release in the presynaptic terminals and leading to the motor impairments causing Parkinson's disease. So, it is known that alpha-synuclein preformed fibrils are more toxic than its monomer. So, how are these aggregated proteins are handled? So, there are different protein degradation pathways. One such pathway is ubiquitin proteasomal system. Ubiquitin proteasomal system is a selective protein degradation pathway. It breaks down the short-lived soluble misfolded proteins which are tagged by ubiquitin in the 26S proteasomal in the 26S proteasome. But ubiquitin protease system does not handle the aggregated proteins, which is handled by autophagy and lysosomal pathway. It is less specific but handles the bulk degradation, which involves misfolded proteins, protein aggregates, and damaged organelles such as mitochondria, which is called as mitophagy, and peroxisomes such as peroxyphagy. So it has several stages where isolation membrane or phagophore is formed around the aggregated or misfolded proteins and dysfunctional mitochondria or any damaged cell organelles, forming an autophagosome. This autophagosome fuses with the lysosome and forms autolysosome, where this cargo or debris is degraded or sequestered inside the lysosome. So, autophagy dysfunction has been implicated in major neurodegenerative diseases such as Alzheimer's disease and Parkinson's disease. So, let us see what are the available treatments for Parkinson's disease. Well, there is no known cure for Parkinson disease or any known treatments that can slow down the progression of disease. Treatment of treatment for Parkinson disease is purely focused on controlling the symptoms of PD for as long as possible by reducing side effects and improving the quality of life of the patients. So there are several drugs which are used to help the manage the symptoms, like motor symptoms of Parkinson's disease. One such effective therapy is by carbidopa levodopa therapy, which is used to treat the motor symptoms of Parkinson disease. So here, the another available treatment is deep brain stimulation, which is helpful in the advanced Parkinson disease stages, where an electrode is implanted and it acts like a brain pacemaker, where it sends electrical pulses to specific brain region, reducing the motor symptoms like tremors, stiffness, and slowness. So it is very important to focus on disease modifying strategies to reduce synuclein aggregates. So there are some small molecule drugs that target alpha-synuclein aggregation such as ANLE 138B, PBT 434, and NPT are still under clinical development, and they recently crossed the phase 1 clinical trials. So, current research seeks to find ways to stop the progression. In this regard, small molecules that inhibit the aggregation of alpha-synuclein and inhibits the ubiquitin-specific proteases and enhances the autophagy are important to mitigate the alpha-synuclein toxicity. So now, what is the rationale behind our study? So our study is driven by a key observation from Sadio S group, where they have identified strongest modulators of oxygen consumption, which is the amount used by the body. So they have identified strongest activators and inhibitors of oxygen consumption in a high throughput screen of 1600 compounds, and they have identified activators at 2 hours and 24 hours. Among these, we focused on the drug nitazoxanide, which is a broad spectrum anti-helminth drug and antiviral and antiparasitic drug, which is approved by the US for treating diarrhea. And not only that, it has an excellent safety profile, which was used by over 500 million people worldwide. Not only that, the recent studies investigated that it can be very well tolerated up to 1500 mg twice daily. So our earlier findings demonstrated that nitazoxanide is an mitochondrial uncoupler, and interestingly, some other reports are also identified NTZ as an activator of autophagy. So let me tell you what is mitochondrial uncoupling. So mitochondrial uncoupling can be defined as a dissociation between mitochondrial membrane potential generation and its use for mitochondria dependent ATP synthesis. As we all know that oxidative phosphorylation and ATP synthesis, both of them are tightly coupled process. So whenever there is mitochondrial uncoupling, so there won't be any ATP production. So initially, this mitochondrial uncoupling was perceived as a dysfunctional mitochondria, but later the discovery of uncoupling protein indicated its potential involvement in various biological processes, such as the proton leak occurring during mitochondrial uncoupling encounters the ROS generation, and it activates autophagy to reduce the energy stress or burden, and it helps in the mitochondrial turnover, such as mitophagy. It lowers the ATP production and increased oxygen consumption. In this regard, we have asked our first question, that does NDZ induced mitochondrial uncoupling restore PD mimetic induced dysfunction in autophagy? As I already told, there are various PD mimetics in uh, like rotinone, MPTP, and 6-OHDA. For this study, we have chosen MPTP in the in vivo and MPP in vitro model. So let me tell you an interesting story behind this MPTP discovery. So in the early 1980s, a group of young drug users suddenly developed severe Parkinson-like symptoms almost overnight, which is impossible because, as we all know, that Parkinson disease or neurodegenerative diseases are age-related diseases. So scientists were astonished and they traced it back to a contaminant called MPTP, which was accidentally produced during the synthetic heroin preparation. So this is what they were using. So it was contaminated with the MPTP. Once inside the MPTP, sorry, inside the brain, this MPTP is a highly lipophilic compound which crosses the blood-brain barrier and converts to MPP, that is methylphenylpyridinium. And after entering the brain, MPP inhibits the complex 1 of mitochondria and enhances the oxidative stress and increases the alpha-synuclein aggregation. So MPP selectively kills dopamine neurons in the substantia nigra, just like in the Parkinson's disease, which is remarkable. So this shocking discovery became a breakthrough, giving researchers one of the most powerful models to study the Parkinson's disease. What began as a tragedy became a great scientific breakthrough, revolutionizing how we study and understand the Parkinson's disease. So here, so we sought to test, as earlier we have reported that NTZ enhances oxygen consumption rate in dopaminergic cells. So to confirm this in a more suitable cell line model, we have used human dopaminergic IMA32 cells, and here we have performed mitostress assay. This assay is a well recognized assay for measuring mitochondrial function or mitochondrial respiration, which means the mitochondrial respiration is nothing but a metabolic process where mitochondria consume oxygen to synthesize ATP through oxidative phosphorylation. So this mitostress assay is uh, where it uses different mitochondrial modulators such as oligomycin, which is an ATP synthase inhibitor. So whenever we add this oligomycin, there will be drop in oxygen consumption rate, giving us to estimate the ATP production. So FCCP is an known mitochondrial uncoupler. Whenever we add this, there will be increase in the maximal respiration. And to shut down the mitochondria, we have used rotinone, which is a complex 1 inhibitor. And there is one more mitochondrial inhibitor, that is complex 3 of mitochondrial inhibitor, that is antimycin A. So here we have performed a mitostress assay where to IMR32 cells we have recorded the basal respiration, and at this point we have added NTZ. As what we can see, there is an increase in the oxygen consumption rate in the cells when compared to the control. So here, after the addition of oligomycin, there is a sharp drop in the oxygen consumption rate, giving us the ATP production levels. Interestingly, we have found a significant increase in the proton leak in the NTZ treated group when compared to the control. So proton leak is a process where protons move back across the inner mitochondrial membrane into the matrix without driving ATP synthase for the ATP production. Just bypassing this ATP synthase, they could increase the mitochondrial respiration in the cells. So by this experiment, we have confirmed that NTZ is a mitochondrial uncoupler and enhances the mitochondrial respiration in cells. As most of the mitochondrial uncouplers are known to counteract the ROS generation, we then move to check ROS generation in the cells. So here subsequently, IMR32 cells were treated with MPP for 24 hours, and as what we could see from the images, there is a substantial increase in the cellular ROS levels when compared to the control. However, when the cells were pre-treated with the NTZ, the MPP-induced oxidative stress was substantially reduced. So here we also performed an experiment to check whether NTZ has the potential to restore respiration in mitochondria that were pre-incubated with the MPP. As I already told, MPP is a complex 1 inhibitor, so in which respiration is completely ceased. So here we have recorded the basal respiration in IMR32 cells, and later, we have added MPP at this point and monitored the oxygen consumption rate for 2-hour time point. As what we could see from the image, there is no oxygen consumption rate or negligible oxygen consumption rate in the cells treated with the MPP when compared to the control group. So interestingly, at this point, when we have added NTZ, there is an immediate increase in the oxygen consumption rate. So here we can clearly say that the NTZ restored the respiration in partially damaged mitochondria by MPP. So this experiments clearly shows NTZ not only suppressed MPP-induced ROS generation in the cells, but also the mitochondrial dysfunction by MPP. So volume of results or references have shown that mitochondrial uncouplers can activate autophagy. As what we can see in this particular paper, that cloxyquine uncouples mitochondria and induces autophagy. And in this particular paper, they shows induction of autophagy by depolarization of mitochondria, nothing but mitochondrial uncoupling. Not only that, interestingly, other reports are also identified NTZ as an activator of activator of autophagy. So here we then asked a question, as NTZ is able to enhance the mitochondrial uncoupling and decrease the oxidative stress, and NTZ can activate autophagy through different studies. Here we have asked a question, how does whether NTZ activated mitochondrial uncoupling activates autophagy or not. So in this regard, there are various studies showing AMPK is activated in response to energy stress or metabolic disruption or mitochondrial dysfunction by sensing AMP is to ATP ratio, and thereby restores energy balance by restoring the or regulating the catabolism or anabolic pathways. So here activated AMPK promotes the catabolic pathways and inhibits the anabolic pathways. So we are very much interested in the AMPK mediated autophagy lysosomal function related mechanism by nitazoxanide. So We next move to check whether NTZ induces autophagy in the cells or not. So here, P62 and LC32 are the markers of the macroautophagy. So the conversion of LC31 to LC32 by lipidation, followed by degradation of LC32 bond cargo in the lysosomes, are the hallmarks of macroautophagy. And P62 is an adapter protein which binds to the cargo. So if there is an induction in the autophagy, there will be increased LC32 levels and decreased P62 levels, and the autophagy flux is increased. If there is early suppression, LC32 levels are decreased and P62 levels are increased. That means in early suppression, there won't be autophagosomal formation at all. That is the reason LC32 levels are decreased. In the late suppression of autophagy, the LC32 levels and P62 levels are increased and flux will be decreased. So as what we can see, in the late suppression, autophagosome does not fuse with the lysosome, thereby accumulating the LC32 bound autophagosome in the cells. So let us see. Here we have treated IMR32 cells with nitazoxanide, two different dosages, that is 5 and 10. As what we can see from the immunoblotting results, there is significant increase in the LC32 levels with concomitant decrease in the P62 levels. So this experiment clearly says that NTZ induces macroautophagy dose dependently in the cells. So next we move to check autophagy flux in the cells. Why it is important to check the autophagy flux in the cells, and it is often defined as a measure of autophagic degradation activity. And there are a number of methods currently utilized to assess autophagy flux. And one such mechanism or method is like by adding inhibitor and activator together to the cells. Here we have as what we can see, the treatment is activator. If there is activator, there will be more LC32 levels. So when we add inhibitor and the activator together, there will be accumulation of LC3B2. As I said in the earlier slide, there won't be any fusion of autophagosome with the lysosome, so that there will be accumulation of LC32 levels. So for this, we have uh, used bafilomycin. Bafilomycin A1 is a commonly used autophagy inhibitor to measure autophagy flux. So bafilomycin inhibits the later stages of the autophagy. So for this, we have treated IMR32 cells with NTZ in presence and absence of the bafilomycin. As what we can clearly see here, NTZ treated group significantly decreased P62 levels with concomitant increase in the LC32 levels. So interestingly, in the bafilomycin and NTZ treated group, there is significant increase in P62 and LC32 levels when compared to the only NTZ. So this clearly shows that NTZ enhances autophagy flux in the cells. Now we asked whether AMPK activity is increased or not, as mitochondrial uncoupling tends to increase the AMPK activity. So there are various pathways influenced by various pathways for autophagy activation, and one of the such pathway is inhibition of mTORC1, regulation of ALK1, or activation of FOXO and JNK pathways. So let us see whether NTZ activates phospho-AMPK or not in the cells. So the treatment with NTZ for 24 hours significantly increase the phospho-AMPK levels in the cells, which clearly shows that NTZ activates AMPK pathway. Not only that, it also significantly enhanced the phospho-JNK levels in the cells. To confirm whether the AMPK pathway is involved in NTZ-induced autophagy, we have used compound C, which is an inhibitor of AMPK. So here we have treated IMR32 cells with NTZ in presence and absence of the compound C. As what we can clearly see here, there is a significant increase in the phospho-JNK levels in NTZ treated group, and which is uh, inhibited by compound C and NTZ treated group. So not only that, we have also observed significant increase in the P62 and LC32 levels when compared to the only NTZ. So this clearly shows that NTZ induces autophagy by activating AMPK and JNK pathway. Now we are interested to check whether the PD mimetic MPP inhibits the cellular autophagy or not. So there are previous reports indicated that the MPP inhibits the initial stages of the autophagy, particularly the formation of autophagosomes in the cells. So here we have treated cells with MPP with 1 and 2 millimolar concentration for 24 hours, as the immunoblotting results demonstrates that there is a significant increase in the P62 levels with concomitant significant decrease in the LC32 levels, which clearly shows MPP inhibits the initial stages of autophagy in the cells. To further confirm, we have performed immunofluorescence assay by using LC3B and LAMP1. LAMP1 is lysosomal membrane protein, and here the co-localization of LC3B and LAMP1 were clearly visible in the NTZ treated group. And in MPP treated group, as what we can say, there is clearly substantial decrease in the LC3B staining, and uh, which is cleared in presence of NTZ and MPP. So to confirm this, we have also utilized another detection method for autophagic flux by using EGFP LC3 plasmid. So here we have used HK293 cells to overexpress this particular plasmid. So as what we can see after the overexpression, when we have treated with MPP, there are no autophagosomal formation or decreased autophagosomal formation in the MPP. So here we have treated chloroquine, which is another autophagy inhibitor. As what we can clearly see, there is an accumulation of autophagosomes in the chloroquine treated condition. We then move to further check it in immunoblotting, where the immunoblotting result clearly shows the MPP treated group substantially decreased the LC3B levels, which were restored by NTZ addition. So by this, it is clear that NTZ mitigates MPP mediated dysfunction in the cellular autophagy. Till now, we have seen NTZ induced mitochondrial uncoupling activates AMPK, JNK signaling pathway and induces macroautophagy in the cells. And now we question whether activated autophagy could clear the alpha-synuclein toxicity in the cells or not. In this regard, we have treated the cells with NTZ different concentration, 1, 5 and 10 micromolar. As what we can see here, there is a dose-dependent decrease in the alpha-synuclein levels in the cells, which is significant. Not only that, we wanted to confirm the MPP role on accumulation of alpha-synuclein. So here we have treated cells with different concentration of MPP, that is 1 and 2 millimolar. As what we can see, there is a significant increase in the alpha-synuclein expression treated with MPP. So now we wanted to check whether NTZ mitigates this MPP mediated alpha-synuclein accumulation or not. So to this, we have treated cells with NTZ in presence and absence of MPP. So there is a significant increase in the alpha-synuclein levels when compared to the control, which was mitigated by pre-treatment with NTZ in NTZ plus MPP group. So now we wanted to check whether AMPK pathway is involved or not in this particular condition. So we have treated cells with nitazoxanide in presence and absence of compound C, which is an AMPK inhibitor. So here, as what we can see, the result, there is a significant decrease in the alpha-synuclein level, interestingly, which was where here NTZ could not decrease the alpha-synuclein levels in presence of the compound C, which clearly indicates that it is the autophagy activate NTZ clearance of alpha-synuclein by autophagy requires AMPK pathway. So now we asked a question: does increased AMPK activity dependent on the mitochondria or not? So for this, uh, as most of the uncouplers of mitochondrial respiration are capable of inducing autophagy via the AMPK pathway, we have examined the effect of NTZ on autophagy in cells lacking functional mitochondria. So to examine this, mitochondrial DNA depleted IMR32 cells, which are called as row 0 cells, were generated by exposing the cells to lower concentration of ethidium bromide for 4 weeks, and the loss of mitochondrial DNA was confirmed using mitochondrial DNA specific primers. So we have already confirmed the cells are devoid of functional mitochondria by this experiment, and now we have performed mitochondrial respiration assay to confirm the role of NTZ on mitochondria lacking cells. So as what we can see here in the normal cells, when we have added NTZ, there is an immediate response to the NTZ, and there is a significant increase in the oxygen consumption rate when compared to the control. But in the row 0 cells, where the cells are non-responsive to the nitazoxanide, which shows that functional mitochondria are essential for NTZ mediated mitochondrial respiration. Now we wanted to confirm whether uh, NTZ requires functional mitochondria for activating autophagy and activating for clearance of alpha-synuclein or not. For this, we have treated this row 0 cells with NTZ in presence and absence of the compound C. As what we can see from this particular experiment, the alpha-synuclein and P62 levels are significantly decreased in the row plus cells, but which are equal to control in the row 0 cells. So this obtained results clearly shows that functional mitochondria are essential for nitazoxanide mediated clearance of alpha-synuclein by autophagy via the AMPK pathway. So here, till now, we have seen the endogenous levels of alpha-synuclein was cleared by NTZ. As I already mentioned, preformed fibrils of alpha-synuclein are more toxic when compared to the monomers. So we have performed another experiment where we have used a preformed fibrils of alpha-synuclein from recombinant alpha-synuclein monomers. Here, we have used a full-length wild-type alpha-synuclein, which is histidine tagged one, and prepared alpha-synuclein preformed fibrils from recombinant monomeric his-tagged alpha-synuclein protein, where we have incubated this protein for 5 days at 37 degrees centigrade on continuous shaking. And later we have performed THT fluorescence assay. As what we can see clear here, there's significant increase in the THT fluorescence with the PFF showing the protein is aggregated. And then we have treated cells with NTZ in presence and absence of alpha-synuclein PFFs for 8 hours and 24 hours. And what we observed was the remarkable results, where we can see at 8 hours and 24 hours, there is an increased alpha-synuclein levels, which are significant when compared to the control. With not only that, under NTZ and alpha-synuclein treated condition, we can see decreased alpha-synuclein levels concomitant with the increase in LC32 levels, which shows that NTZ promotes the clearance of preformed fibrils of alpha-synuclein in cells via autophagy. So this clearly states that NTZ induced mitochondrial uncoupling activates AMPK JNK signaling pathway, induces autophagy, and enhances the alpha-synuclein clearance in the cells. Now, we wanted to demonstrate the efficacy of nitazoxanide in animal model of Parkinson's disease. So here we have opted for subacute MPTP model, where MPTP was given to mice for 5 days. So in our experiment, we have pre-treated the mice with NTZ with an oral dosage of 50 mg per kg body weight continuously for 10 days. And later, from the second day, we have given MPTP intraperitoneally 20 mg per kg body weight, and later on we have observed the behavioral test. So here, why we have the importance of MPTP, as I already mentioned, it clearly decreases the TH positive neurons in the substantia nigra pars compacta region, also in the striatal region. So what is this TH? TH is tyrosine hydroxylase, which is a rate limiting enzyme that converts tyrosine to L-DOPA. So this L-DOPA crosses the blood-brain barrier, and it is a precursor for the dopamine, which is essential for motor coordination. So this is essential for this TH is essential for the dopamine production. So in Parkinson's disease, reduced TH expression activity in the striatum and substantia nigra leads to dopamine deficiency. So loss of TH producing neurons are the hallmark of PD pathology. So here we have performed immunohistochemistry analysis at SNPC regions of mice. As what we can clearly see, this is the SNPC region, and there are like more TH positive fibers and neurons in the control, where there is significant decrease or loss of TH positive neurons in the MPTP group. So which is restored by the NTZ pre-treated group. As what we can see, the TH positive fibers are significantly increased or enhanced in this group when compared to the MPTP group, and as such the NTZ alone group is almost similar to the control. So not only in the SNPC neurons, similar results were found in the immunohistochemistry of striatum sections, where in the NTZ plus MPTP group has demonstrated a substantial increase in the TH positive staining when compared to the MPTP. So to further validate this, we have performed immunoblotting analysis on SNPC tissue and validated the alpha-synuclein levels and TH levels in this tissue. As what we can see here, in MPTP treated group, there is a substantial decrease in TH levels in concomitant with the increased alpha-synuclein levels, which was restored in the MPTP plus NTZ treated group, where we can see substantial increase in the TH levels and decreased alpha-synuclein levels. So this clearly shows that NTZ restores MPTP mediated loss of TH nerve fibers in the striatum as well as in the substantia nigra pars compacta region of the brain. So in order to assess the physical activity and locomotor coordination of mice, a series of behavioral experiments were conducted when compared to the control group. Series of behavioral experiments were conducted to know the motor coordination in the mice. So here, when compared to the control group, the MPTP group displayed significant reduction in the locomotor activity, not only in the locomotor activity, but also in the rotor rod and grip strength test. However, the motor activity loss induced by MPTP group was significantly prevented by pre-treatment with the NTZ, which is significantly high when compared to the MPTP. So similar trend of results were observed in rotor rod and also in the grip strength test. So all the above findings collectively indicate that NTZ ameliorates motor impairments in the MPTP mice model of Parkinson's disease. So here, oral administration of NTZ conferred significant protection in subacute MPTP mice model of PD. Not only that, NTZ per se substantially reduce the levels of alpha-synuclein and enhance the TH levels, and MPTP mediated alterations in alpha-synuclein and TH levels were ameliorated by NTZ treatments. So taken together, all the above observations highlight the potential of NTZ in the treatment of Parkinson's disease. So moving to the summary, so the mitochondrial uncoupling nature of nitazoxanide enhances the uh, nitazoxanide increases the AMP to ATP ratio, where ultimately activating phospho-AMPK and JNK pathway, leading to the alpha-synuclein clearance by activating autophagy in the cells. So part of this study was published in Cellular Signaling Journal, and these are other publications from my group. And I would like to acknowledge Dr. Bonnie Firestein, my present mentor, and I'm always indebted to my PhD boss, Dr. Shashivardhan Kalivendi from IICT, and I thank my friend and present collaborator, Dr. Mayank Kumar Singh, and SPSR team, especially Dr. Shashi Alok, and the rest of the team for this opportunity, and all lab members and friends in University of Kentucky and in IICT, and especially my family, my lovely daughter Laukya and my husband. Always, they have always supported me. And I would like to end my session here with a cartoon on Parkinson's disease. So this looks little comedy, but it is a hard reality. Like most of the Parkinson patients spill whatever they hold. So that's it. And thank you. And I'm happy to take the questions.
Thank you very much, Dr. Niharika, for delivering such an insightful and scientifically enriching lecture on role of nitazoxanide mediated mitochondrial uncoupling in the clearance of alpha-synuclein by autophagy, implications for Parkinson's disease. Your presentation provided a deep understanding of the complex mechanism underlying neurodegeneration, particularly the role of mitochondrial dynamics in autophagy in clearing alpha-synuclein. The way you connected fundamental molecular insights with potential therapeutic strategies was truly impressive and highly valuable for both researchers and students. On behalf of the Society of Pharmaceutical Sciences and Research, we sincerely appreciate your valuable time, expertise, and contributions to advancing knowledge in this important area of neuroscience research. Your talk has been truly inspiring and impactful for all the participants. Thank you once again for being the part of 106th SPSR International Webinar.
Thank you
Now
so
there are
much, Dr. Pradeep.
Thank you so much, ma'am. Now there are some questions from the participants, and
Sure.
I will be asking you one by one. The first
Yeah.
one is, ma'am, why is autophagy considered a key therapeutic target in neurodegenerative disorders like Parkinson's disease?
So, as we see in the neurodegenerative diseases, the major concern is the aggregation of the proteins. Like in Parkinson disease, it is the aggregation of alpha-synuclein, and in Alzheimer's disease, it is the aggregation of tau and amyloid. In Huntington's disease, it is Huntington's protein. So, these aggregated proteins are well handled by the autophagy lysosomal pathway, because no other pathway clears these aggregated proteins, such as like UPS, as I already mentioned in the my earlier slides. It only handles the misfolded smaller proteins rather than aggregated proteins. And not only that, in most of the patient brains or PD or AD brains, it is known that the autophagy is one of the autophagy gene, that is ATG7, is deleted, leading to the like leading to the neurodegeneration in the mice studies. So it is very important to target autophagy if you want to clear the aggregated proteins.
Right, and thank you so much.
Yeah.
The next is, ma'am, how does nitazoxanide mediated mitochondrial uncoupling differ from pathological mitochondrial dysfunction?
So, nitazoxanide mediated mitochondrial uncoupling differs a lot because usually we have done an experiment in my previous initial studies where we wanted to confirm whether really nitazoxanide is well tolerated or not. For this, we have seen some of the cytotoxic assays. To the cells, it is very well tolerated up to 50 micromolar. I haven't seen death at 100 micromolar concentration also. Not only that, so this mitochondrial uncoupling uh, stays in the cell when the nitazoxanide is present in the medium. So whenever we washed away the medium, so the cell goes back to the normal condition without any proton leak or something. So this mitochondrial uncoupling nature of nitazoxanide is transient. And not only that, nitazoxanide has a very short half life, that is like 8 hours, I think. Uh, like that is a good point for the nitazoxanide to be repurposed as a drug.
Right, ma'am. Thank you so much. The last one is, could mitochondrial uncouplers have potential side effects, and how can their safety be optimized?
Yes, there are some mitochondrial uncouplers like FCCP and CCCP. They are very strong mitochondrial uncouplers, and their activity is not reversible. So, initial days, I think DNP, that is 2,4-DNP dinitrophenyl, was used for weight loss, which was later on banned by the US FDA drug, which caused uh, like uh, which is lethal to the patients. Now there are many mitochondrial uncouplers which are mild, like BAM15 or liver targeted form of 2,4-DNP, and they they have designed in such a way that they particularly target only uh, the target specific areas, so that these side effects are uh, decreased or well handled.
Right, ma'am. Thank you so much for Dr. Niharika for your insightful and comprehensive responses to all the questions. Your
Thank you.
explanations have significantly, thank you, ma'am, deepened our understanding of complex mechanism underlying Parkinson's disease, particularly the role of mitochondrial uncoupling, autophagy regulation, and alpha-synuclein clearance. The way you connected the fundamental research with potential therapeutic implications was truly remarkable and highly inspiring for both the students and researchers. Thank you once again for your excellent contribution to the 106th SPSR International Webinar. So with this, we come to the end of 106th SPSR International Webinar. Today's session on role of nitazoxanide mediated mitochondrial uncoupling in the clearance of alpha-synuclein by autophagy, implications for Parkinson's disease, has been highly insightful and intellectually enriching. We sincerely thank our esteemed speaker, Dr. Niharika Reddy, for delivering such an outstanding presentation and for sharing her valuable research findings with us. We also extend our heartfelt gratitude to all the participants for their active participation, thoughtful questions, and continued support, which made this session truly engaging and successful. We are deeply thankful to Dr. Shashi Alok, the National President of SPSR, for honoring us with his presence. We recognize and appreciate your commitment and guidance, and we offer our profound thanks for your significant input. We would also like to express our appreciation to our convener, Mrs. Monika Sabharwal, ma'am, for her pivotal role in organizing this event. We extend our gratitude to all the SPSR team members for their collaborative efforts, which have broadened our understanding and fostered cooperative learning. Your collective contributions have made this webinar a success, and we look forward to the continued contributions, collaborations in advancing pharmaceutical sciences and research. With this, we formally bring the session to a close. We look forward to welcoming you to our future SPSR webinars and academic initiatives. Thank you and have a wonderful and productive day ahead. Thank you so much, ma'am. Thanks from Team SPSR.
Thank you so much, and SPSR organizers and team for this wonderful opportunity.
Thanks, ma'am.